to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 86th episode of the Weekly Bioanalysis, the official podcast of KCAS Bio. KCAS Bio is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. My name is John Perkins and I've been in the bioanalytical LCMS space for around 30 years. I'm here with my co-host, Dominic Marino.
Hi, my name is Dominic Marino, and I've likewise been in the bioanalytical space for close to 30 years.
Dom and I are members of the growing scientific advisory team at KCAS Bio. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS Bio's services. Today we're here with a guest, Dr Cheikh Kane, Vice President, Biopharma Services at KCAS Bio.
Thank you, John and Dominic, for having me. I look forward to this discussion.
We are thrilled to have you listening to the 86th episode of the podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcasts, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining us for the first time or if you're a regular listener. Today's podcast will be a review of the latest news and resources and then a focus on the topic of our choosing before discussing any feedback we've had from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we are excited to have you here and we are looking forward to a fun episode today. To kick us off for podcast number 86, Dominic is going to go over today's podcast topic a bit before we jump into the news and resources section. Dom.
Yeah, we got a jam-packed podcast today. Should be incredibly exciting. Got a great special guest in Dr. Kane. He'll give us a little intro and background and what his role here is at KCAS Bio. Then we're going to dive right into immunogenicity, what it is. We're going to talk about some standard approaches for ADA. We're going to introduce you to the lean ADA approach. And then we're going to have more of a discussion around when it's suitable to take the lean ADA approach and maybe some perspectives from the industry on the lean ADA approach. So, John, we got a jam-packed episode here. Without further ado, I'll kick it back to you to get us started. Always, we love a good GLP talk. John, tell us a little bit about this.
Yeah, we can't get away from GLP-1s at the moment. This is really interesting. I have some anecdotal backup to this. Basically, the headline is Novo's weight loss drug helps curb some drinking habits, small study shows. And this was a study done at the University of North Carolina, Chapel Hill, where they found that Wegovy might actually help with curbing alcohol consumption. It was a small phase 2 study, and basically they found that it affected alcohol cravings and the number of drinks consumed on drinking days. But it was like the number of, as far as I interpret it, the number of alcoholic drinks on drinking days. But the overall, they didn't stop them drinking other drinks. And so it was like patients with alcohol use disorder, they either had 1 weekly semaglutide or placebo over 9 weeks. Those who received Novo's drug got the lowest dose during the first 4 weeks, then increased to 0.5 mg per week for weeks 5 through 8, then got up into 1 mg. And they found that the proportion of patients who were given semaglutide and reported zero heavy drink days increased significantly over the 2 dose phases. But the drug did not appear to alter the proportion of abstinence and drinking days. So it's like, it's almost like it doesn't stop you, the days you don't drink, it doesn't make you want to drink when you're not drinking, but it does stop you drinking when you are drinking. And the anecdotal evidence that is, my wife has actually started with Manjaro. And we like to have a glass of wine in the evening, and both of us have been talking for a long time. Yeah, it'd be really good if we didn't drink quite so often. But what we find is that she really does that sort of, that consumption really goes down.
Yeah.
So we've seen it just domestically, but here you have a phase 2 study pointing to an actual longer term application for the GLP-1 drugs.
Yeah, it's funny they say it doesn't like thirst, right? That it doesn't diminish your desire to drink like water, for instance. That's what they're kind of alluding to.
That's the way I interpret it. I may be wrong.
No, I think that's it. Because you don't want to scare people off from taking something that could prevent you from even smoking, right, and drinking. But it makes sense, right? Like your desire for hunger is the same as the thirst pathways, all of that's connected. You don't have two separate systems for, when your stomach's growling, you might just be thirsty, right? And so it's not that surprising. And I do have a friend that's on Wegovy, and he substantially drinks less now. In fact, he doesn't feel great when he does drink, and that might be part of it too. It's just so the desire is not there. But it's a fascinating study. It's a wonder drug. I think my add to this is just like I think it's, when I say wonder drug, right, it just seems to have a lot of ability to, just got a lot of utility to it. It's flying off the shelves, right? There was a supply, John. I don't know how much you've been following it, but like the supply chain issue with it was on, people could make like almost generics of it due to supply chain issue, and that's becoming a big issue now. That compounding issue.
Yeah.
So I just worry about look, take it absolutely. Just make sure you're getting like an approved version, right? Yeah.
So in terms of the compound, but I think it was last week the FDA said that basically the novel drug semaglutide is now, they've resolved the supply issue for that, and they'd already resolved the Lilly one. Yeah. So Manjaro. So in terms of the opportunity for compounders is actually is reduced compared to where it was.
I mean, some of them are fighting it, right? I think it's a hard thing to tell someone who's been selling something for 2 years that this is dangerous and not approved when there's, probably thousands of people that have taken your medication. But it's a fascinating study, John. I didn't realize it was going into a phase 2. Really cool stuff. I mean, but it all kind of makes sense that metabolism's all kind of linked, right? That's what I think of this.
Yep. And one thing I didn't touch on is, like it says, a smaller subset of patients that were smoking cigarettes before the study found the medication by time reductions in cigarette smoke per day as well. So really, it's like you say, it's fascinating in terms of those cravings that it sort of seems to tamp down on.
Well, John, maybe we can move on after this, but the one worry is I still worry about like a healthy 20-year-old taking this, or a healthy 30-year-old, but it seems to be fine. I mean, heck, I've been considering it and all this, but I'm sure your wife's taking it, but like we're just a little, our metabolism is senescing, and it kind of makes more sense to maybe do something. I just worry a little bit about
I mean, it's like, but if it's healthy 20 or 30-year-olds, I mean, they shouldn't be taking it because like, why would you take it?
They are, right? Like,
if you're a healthy functioning individual, I mean,
we're a vain society, unfortunately. There are a lot of people taking it.
Understood, but I mean, I suppose that's the thing where it's looking for these different routes beyond the physician, because at least for us, it was done in consultation with a physician. So a healthy younger person shouldn't be allowed to pick it up through that route.
It's definitely changing lives. It seems to be.
Oh, sure, absolutely.
Over the last 3 to 4 years, it's got to be one of the biggest things to impact public health.
Mm-hmm.
But it's not going anywhere, and
I think it's what's intriguing living with it on a daily basis is that the reminder to make yourself eat type of thing. I know one of our colleagues talked about that they don't feel the need to eat.
Yeah.
And it's like, so you have to remind yourself that you have, because that's a balance you have to get right, because you definitely don't want this drug's not like, if it's sort of reducing your want to eat, you don't want to go too far in the other direction, because that's unhealthy as well. So.
Exactly. You start, if you don't know, John, you're right. There, you can be too skinny. It really taxes your kidneys if you start breaking down.
Yeah. And so you find that at the moment I'm in that mood where, because I go through phases, I'm in the mood right now to cook stuff. So of course I'm cooking all this food, and then it's like I eat most of it because I'm the one with the appetite, which is fine. I'm not complaining on that. I find I'm still giving a lot away to because my daughter lives around the corner, giving her like lots of stuff. It's like I made a big pie the other day and sent a quarter of it off with her, because she stopped off for some stuff. I was like, have some pie, take it.
I throw it away, John. We can move on. I throw more food away because of what you just said. I got zero desire to save it. I don't take things home from restaurants like I used to. I don't know. But that is definitely something that as you get older, you learn that and then it's hard to cook for two versus 4
Sure.
or 5, whatever. But
Well, I think, well, for that, it's the fun of doing different things for
yeah.
me. That's why I'm doing it,
Oh no, I
So,
still love cooking. I just freeze it more. Like I just
sure.
take it and
Yeah.
free. Like even when I make a recipe, I tend to think, okay, I'm gonna vacuum seal 2/3 of this and make 3 meals out of it. But
Yep.
John, yeah, good
Let's move
talk
on.
on GLP-1. Yeah, like
Yeah.
real famous scientist here, Nobel
Yep,
laureate.
sure.
This one's about. Yeah, go ahead.
So
Tell
this is interesting because obviously there's a lot of discussion of AI, etc., in the news, and I will be honest, I'm sitting here as a something of an AI skeptic in terms of some of the directions it's being applied, and I'm a great believer. I don't want, like the Microsoft prompt on my when I'm writing a Word document. I don't want its input. I know how I think,
caveman.
and I
We're
know.
caveman, John. We're caveman with that attitude. But go
I
on.
don't.
Yeah.
Yeah, I'm going to stick with that attitude because I know how I write. I know how I want to write, and I know how I will go back through and edit and stuff. I do not want someone else leading me down a path I don't want to go. Anyway, that said, I know there are a lot of other potential applications that could be very valuable, and I thought this was a nice a nice possible application long term. Again, it's very early, but it gives an indication of where AI can be very useful. And we've alluded to this in the past in terms of drug design and stuff. But the headline is David Baker Lab makes de novo enzymes with AI. And so David Baker's a Nobel laureate. And it's like they've looked at designing proteins with computers, and now taking it a step further. And some of his researchers, I should find, yeah, it was a trio of scientists, Anna Lauko, Sam Pellock and Kiera Sumida. They want to make new hydrolases. So basically, that's sort of using the enzyme to obviously cut down molecules, etc. And these are enzymes that have been found across nature that get cue up plastics, helping muscles contract, relax, and more. And the thought is long term, this might help with some of these, therapeutic approaches that require enzymatic cuts, things like that, and they may have application with that in the future. I really like and it's really interesting because this team sort of said we were going nowhere for a long time, and then one of the other groups, one of other David Baker's group, came up with another different approach called RF diffusion that really then helped them really trigger and boost their making artificial enzymes using or design comparative enzymes using AI. So I'll pass it back to you.
No, I think that last piece is just two heads are better than one, and
Yeah,
we're
absolutely.
better than two, and it's part of why we're set up with like an LBA group and a protein mass spec group to kind of the more we talk, the better the solution there. But AI is an interesting thing. There are some others. I feel like some of them are in clinical trials. They're purely designed from this. Um, you know, David Baker is world famous, right? I feel like he's from the 90s. I think he was the first to help with the Genome Project. I feel like maybe that was his company. Got probably some of the most brilliant minds in the world working on this, right, John?
Yep.
And the target they've got is great in
Mhm.
terms of like new enzymes, and kind of seems like it's getting into that CRISPR space somewhere in there a little bit, right? Some of the gene therapy space. And then I feel like the plastic thing was really interesting. That probably
Sure.
has the most. But the AI in our lifetime has a lot of application, and I think it's to help like not only identify these things, but you still gotta go do it, right?
Yeah, absolutely.
And
I
and,
mean,
and
I
but
think
we can trim that down, right? So I mean, I think the whole concept here is like it's not like bioanalysis is going away. I don't or
no,
drug
no, no.
development, but and I don't know, maybe in another 100 years, who knows? But the applications there are great with obviously what you just talked about, designing novel therapeutics. And then I do think it has good application with maybe reducing the number of like, preclinical trials and things
Sure.
like that.
Yeah. And
And then we have
yeah,
another use too, right? I
there's
do think it has big use in bioanalysis, but that's like its own separate kind of podcast, John, right? We can talk about that at
I
a
think
different time. Yeah,
I
go ahead.
think for this is the hope is you can design things quicker, more efficiently, more intelligently, that will lead you to a drug that has more of an application. Like you say, that leads to, if you can have a honed process, you reduce your number of clinical trials, things like
Yeah.
that, and the number of drugs that fail in the process. So that's really the attractive side to this: is can you use all this computing power to really help drive you quicker to solve a medical problem.
Right.
I'm going to sort of give it some of the group's take on this, and I think, because I think this is interesting. It's like the AI made enzymes aren't as active as the best hydrolases found in nature, according to the lab results. At least for now, nature's outstripping what the group can do. But again, as we all know, it's always like you never crack a problem straight off. It's
No.
always like you build to it. So Loudco said they're still not as good, and that's another future direction, is trying to understand what's missing to make these better and faster. Again, it's all the nature of basic research. You get a result, and it's like, well, it's not quite. It's like me cooking. Actually, it's like I cook things. It's like, yeah, it's not quite how I want it. How can I make this better? It's that's that mindset. But yeah, you're right. They got two major areas they're focusing on: making those plastic degrading enzymes and exploring the therapeutic potential for AI made enzymes with other functions. So their take is proteases or enzymes that make. Proteases are an area to watch. A long-term hope has been able to degrade any protein in the human body by building the right enzyme, which could lead to new, more precise therapies that could also currently unreachable targets. So that's the drive with this. And like I say, it's we've talked about AI design of small molecules, and there's obviously companies focused on that. The Baker team, when I talked about the RF diffusion, that was actually it's a diffusion model that creates proteins. So that, they're obviously very focused in this area, and this is just another step where they're taking it to. I think it's fascinating to see where this goes. So like I say, I'm sort of skeptic on the use of AI, where ultimately it's sort of, why are you using AI? But with something like this, I can totally see where the benefit comes from. You're taking advantage of the computing power to help you come to a solution quicker. That I'm all on board with.
Yeah, I feel like machine learning, even in silico was a term. I feel like we've been doing this for
Mhm.
decades even, but this is just, now it's so at the forefront. And there's still like this kind of dogma about it that it's like AI is somehow kind of evil, and maybe it could be, right? I think that's the piece that scares a lot of people. But for advancements of science, it's here, it's real, and we need to use it. But your skepticism's warranted, John. And I think when you say skeptical, it's like, well, let's just use it for the right things, and don't
Yeah,
overinterpret what you're using it for, right?
yeah, exactly. Yeah, yeah,
But it's
yeah.
good stuff. It's a great. I mean, it's its own topic. It's becoming a group at AAPS, right? It's here for real in the bioanalytical world, and something that we're certainly investing in heavily. But John, let's see. We got a nice special guest today. Maybe we just do this one
Let's
last.
do this one last one, because this is we're always it's always so this. I'll be honest, I'm looking for things to talk about. This was a bad, dull few weeks for news on the pharma side of things.
You didn't want to report all the companies that failed, right?
And I don't want to talk about the mergers and the
Yeah.
drug. I mean, it's interesting to know what's being approved and things like that. But finding those nuggets that are really genuinely, oh wow, there was basically like three topics all came through in one day, and it's like right, we're set.
Yeah.
But this to me is incredible. It's basically the headline: in single case, spinal muscular atrophy drug given before birth spared baby of symptoms. So basically, it's a spinal. There are three spinal muscular atrophy. I think is a rare disease, but there are three approved treatments: Biogen's Spinraza, Novartis's gene therapy Zolgensma, and Roche's oral drug Evrysdi. And they're approved in use. They're approved for people, but not prior to birth. This single case which is this at St Jude's Children's Research Hospital. The family had an elder child who'd had spinal muscular atrophy. But in this case, I think the second child in the womb was showing signs as potential of potential defects. So the mother took Evrysdi, the oral drug. She actually took it in a solution form, which was supplied for free by Roche for 6 weeks before giving birth. The baby appeared normal at birth, but faced birth defects such as heart murmur, which resolved, reduced vision, weakness on the right side of the body, and developmental delays. Researchers said they believe the birth defects occurred early in fetal development before exposure to Evrysdi, and no cause has been identified. But the key to this is once the child was born, they they've exhibited no symptoms of spinal muscular atrophy since. So things like weak muscle tone or lack of certain reflexes. So 8 days after birth, the baby began receiving Evrysdi directly and continues now to take it at 30 months of age. At about 20 months, the baby received gene therapy with Zolgensma, which is approved for children less than 2 years old with spinal muscular atrophy. So this just blows me away.
It does.
I'll pass it back to you, which is why I really want to make sure we touched on it, because this is incredible. It's like, it's like, and like, I know it's a single case, but again, it's like what the potential possibly of doing something like this, but that sort of, you got this oral treatment, administer it to the baby in the womb through the mother, and it's actually helping address some of the symptoms in the womb before then the baby is born, and sort of then eligible or available for further treatment is an incredible thing to do.
John, there's two things that make it like real, like fascinating and incredible, and why we're so like, wow, I can't believe this. One is it's such a sensitive area to treat pregnant moms, right?
Sure.
They have vaccines out there to prevent like bacterial stillborns, and there's all sorts of vaccines that are like basically proven in some respects 100% effective or highly effective, highly safe, but they'll never get approved only because like some of the I don't know if resistance is the right word, but there's just that stigma about, any sort of pregnant woman. It becomes a whole like kind of, it's a little bit taboo to treat, right? Mm-hmm. Although I think there's more and more now. So that's the first kind of thing that made it courageous, right? That word's overused these days, or unprecedented, another overused word. But at first they said, hey, we're going to look at mothers that are pregnant and try to treat them. Now they really did and then to give the mom something to think, hey, that this could be treating the child to prevent it it's just great science.
Sure. Yeah, absolutely. Yeah.
It makes total sense. You can think about it mechanistically why, if they, and John, they didn't, like in vitro, they did not determine if the baby had this, right? They just thought because the brother had it, or did they know?
I think they said, I think if I remember rightly, there was an indication of it.
Okay.
Just let me get it. Keep talking.
So the thought of like taking this oral medication and going across, like, remember the
yes. So here we go. Sorry, I'm going to interrupt, because then you can basically, the family's first child died from the disease, and prenatal testing confirmed the fetus also had SMA, and likely a severe form of the disease. So again, their prognosis was not good. So this is something that this really is. This could be really
a miracle.
We might be able to do something about it.
Yeah, it's a complete miracle. And we've been in science for 30 years. Each of us collectively never heard of anything treating mom, and maybe there are some, but not to this degree of success, right? Where this is a miracle for this family. They were given a death sentence, basically, is what that kind of sounded like.
Sure, absolutely.
And then this very intuitive doctor, hats off to Finkel, is that his name? Just unbelievable kind of, again, courage comes to mind, and even the family and all this, and suddenly they've got a child. It's just, man, this is why science is so great. The pregnancy is still a little bit this is why immunology is so fascinating to me. How that immune privileged area is still kind of kept in the key, the answers to how that takes place, the kind of you got the blood-brain barrier that's one instance that had like your brain's immune privileged, your eyes are immune privileged, right? They just have these mechanisms that are different than your circulating kind of immune system. And the uterus, the womb of a woman is the same kind of we're still trying to understand it, but somebody kind of practically said, hey, this small molecule could probably treat in fetus, right? And that's what happened.
Sure.
So
Yeah.
it's really amazing.
I think it's
Great story.
it's really, the more I look at this, the more like it. There's things that really help them drive this. Like we said, like I mentioned, there's the 3 approved treatments, but the gene therapy Zolgensma couldn't
No.
couldn't. Sorry, Spinraza is currently administered via spinal tap, which is not technically feasible in a fetus. And Zolgensma, they'd actually observed death of fetuses in a sheep model. So neither of those could be. It shows
the oral med seems.
What this does show is that we talk about an indication and first drug to market. There's the one that's going to take lead, and it's like, well, why are we looking for the second, third, and fourth? Going to well, here's a really good reason why you do want those second, third, and fourth, particularly because they're coming from different directions. Having an oral drug and then 2, obviously, large molecule-based therapeutics, it gives you alternative approaches, and one might provide the solution where the others can, which is what happened in this case. I'd like to say, I think there's a lot. The logic in this is amazing, and the whole thing it's such a good case. I saw this. In passing, just on my phone, and then saw a fuller article to it. We have to talk about this because it's amazing.
It is amazing, and it's so on this, like, after you read it, you think, oh wow, that no wonder it worked, right? I mean, it seemed like but coming up with that idea and having the ability to execute on it is, some of the everything. To me, it seems almost simple, John, but there was nothing simple about it, right?
No, not at all. No.
So John, this is a good point to move on.
Exactly.
I want to make sure we get a good discussion in. So let's get going with our main topic. What are we talking about today, John?
So our main topic today, and I'm probably largely going to be on mute for most of this because this is way off into the realm of. So last time we did a lot of talking. This time is way off into the realm of large molecule stuff where I'm like. I like to listen and hear and learn, yes, we're going to talk about immunogenicity. What is a lean anti-drug antibody determination? We have a special guest. We got Dr. Cheikh Kane, who is currently the VP of Biopharma Sciences, at KCAS. And so, thanks very much for joining us, Cheikh. I think the best thing we can do is if you could start by just talk a bit about your background. What brought you to your life through your life? What brought you to Kansas City? Your current role, and we'll take it from there.
Thank you, John. Yeah, Cheikh Kane here. Been within the industry, I would say 2+ decade. Academic background on the microbiology, immunology. A couple of years back, did work on immunobiology for mycoplasma protein. At the time was kind of hot topic in a way. Mycoplasma have this interesting features, right, that have a TGA in their genes that will be coding for tryptophan, but when you go to the universal system, it will be a stop codon, right? Therefore, it's a feature that prevented people in being able to clone full genes into eukaryotic or prokaryotic system. Therefore, we took them now and a goal of determining some of the protein, 3 of them. One of them was the adenosine that will be helping the bacteria to bind to the cell surface, and one membrane protein, and also one cytosolic protein. At the time, we we did really imitation, site-directed mutagenesis on those genes, reconstruct the gene, do sequencing, and reclone them in different type of vectors that will be prokaryotic, eukaryotic, and look into the potential vaccinal ability of that, and also just to understand the pathophysiology of the bacteria. Again, long time ago, right? Been within the industry, as I said, 2+ decade. Started with small biotech, did work also in the big pharma field. At the same time, also works with small and big CROs, right? I would say in the last 15 years or 18, really focus on the bioanalytical field, right? ADA, PK biomarker, and cell-based and NAB assays. But also something very close to my heart also is the really still very involved in the reagent generation and characterization, as I feel it kind of a entry door to the good bioanalytical assays. Why KCAS, right? I think it's very interesting, right? I would say initially when people touch base with me, say, okay, you have opportunity, are you interested in talking about, I said, okay, where? Midwest. I said, oh, no. Right. Sorry. Right. And second time, no. And third time, okay, let me talk. Just and I've been really very surprised, pleasantly surprised, and was kind of a nice ride. At the same time, just on the strategic standpoint, I think was a time where KCAS is trying to get next level, right, in growing, expanding the bioanalytical capabilities and so forth. And it's coincide with also the involvement of the Vitruvian investment, right, within KCAS that was giving us great opportunity in regard to the our overall capabilities, and also gave us this kind of brand new building.
Yeah, Cheikh, I'll brag on you for just a second. You've been an unbelievable partner, teammate over the last few years, and yeah, you're humble about what you've been able to bring to the organization. But that could be its own little topic. But you've, I mean, transformed our biopharma here, and it's just been amazing to watch you come in and grow. Been proud to help you do it, but it's been an incredible growth under you. You mentioned Vitruvian. We got all the tools we need. It is a great work in here. I didn't know you didn't want to come to the Midwest, but that's funny. I could see that, but
I'm totally with you on that one.
Yeah. But no, it's great here, right? So, but we're gonna go right into it. So I have my thoughts on like immunogenicity, right? It's a big word. What is it? Why do we test for it? So I think we have a pretty diverse audience. They've heard the word immunogenicity, but why don't you maybe, because we've talked a little bit about this, right? Maybe we just kind of, if you want, give us a little bit about how we just got to the 3 tiers. They've heard us talk about that, but you can take us wherever we like to go. But I can maybe give you a little bit of like, they were back in the 90s, right? I'm going to go way back, right? That they were human anti-mouse antibodies, right? Human anti-therapeutic or anti-protein. The word immunogenicity haven't even been coined at that time, right? Cheikh, we've talked a little bit about this. But maybe if you just want to give us a little bit of your thoughts on immunogenicity, the audience knows what the 3 tiers are. But you know what, maybe your take, and happy to share mine as well. I got my opinion.
Exactly. I think again, when we say immunogenicity in our field, that is unwanted immunogenicity, right? The immunogenicity per se, it's been for lifetime, right? It's really the vaccine properties of the biologics, right? The ability for a protein when in contact with the cells, right? That without going in detail, present in certain way that trigger the immune response toward that biologic.
Cheikh, I think that's really important. I want to emphasize what you just said. For decades, it was wanted immunogenicity in the vaccine space. It's only until about 10, 15, 20 years ago that immunogenicity became a dirty word. Not that it's a dirty word, but it's unwanted. So I think that's really important to understand that most of what we're going to talk about today is the unwanted immunogenicity response. So thanks, Cheikh.
Yeah, and also it just as that been there, I think it started really when we have the cases that are severe and dramatic cases in the EPO programs, right? It's kind of where synthetic EPO is given to people they developed neutralizing antibodies against the endogenous EPO that end up being provoking the inhibition of the erythrocyte maturation and production. Right.
Yes.
That's
Just because I want to, that's great you mentioned it. It's EPO, right? And that
Mhm.
was used in predominantly, it's still used today to help boost someone's neutrophils, and chemotherapeutic patients take it. But that's the origin of, that's like the hot spot of immunogenicity, because there was a couple things, right? People might have been microwaving it and doing some weird stuff with it that might have caused this, but that ship has sailed, hasn't it? So I want to make sure the audience understood what he's just talking about. He's going back to, like, what was the year? Maybe 95 ish, check.
end of
So,
90s.
so
Late
can
90s.
I ask, as someone completely ignorant, what did they see in patients that what were the
Neutralizing. Yeah, just
what was
so
the reactions,
yeah,
etc.?
so their white blood cell counts aren't coming up. They were
Okay,
taking, yeah,
okay.
that was the neutralizing effect. And it's still kind of not known in check. It was either a combination of storage, and storage could be
Hmm.
people were warming it up, which was
Yep.
really bad, right? Or it might have been the manufacturing. We've come a long ways, like how pure it was, how
Mm.
reproducible it was, are
Sure.
part of the
Yeah.
things that say it's there.
Yeah, and also John, just definitely in addition to what Dominic said, is it was provoking pure red cell aplasia due to the inhibition of erythrocyte maturation and product.
Okay.
Right, and that kind of triggers the focus on the unwanted immunogenicity. I
Hmm.
would say why unwanted immunogenicity, again, it's different, have a different properties that impact that. Right, first one going to be your drug product itself, right, the scaffold.
Yep.
Right,
Yep.
as it get more complex, most likely more complex, more presentation to the T cells, uh, more the likelihood of presenting immune response to it. In addition to that, definitely the formulation of the drug itself will impact also, as it will play a role as an adjuvant.
Hmm.
In addition to the route of immunization, right? Giving a drug product that is a biologic by intravenous versus subcutaneous is very is different in regard to the implication in regard to the human response itself. And then in addition to the route of the immunization, also this type of when you get to the phase 2, again in the situation of the clinical studies or even in preclinical, when you repeat doses, then every single time you're boosting the immune response by that kind of regaining the human response activity, and then that other situations that will create type just a vaccine properties, right, that create human response to the against the target or the
Yeah.
drug product itself.
Yeah, if I'm taking Humira for a couple of years, Shaq, basically every time I give myself that shot of anti-TNF alpha antibody, I'm vaccinating myself, right? I'm actually getting into a setting where I may generate an antibody. But please continue, Shaq.
Yeah,
I feel like we're on a roll.
yeah.
Sure.
I think what is interesting again, in general, why people ask why we test for it, right? The main two implications of the unwanted immunogenicity are going to be the impact of the efficacy of the drug, right? You're giving that drug that is a biologic, and then the animal or human body creating antibody against the drug, right? Which that trigger, it will be in circulation binding to the drug itself, and then it will increase the clearance of the drug, right? Therefore, if you assuming some therapeutic doses, right, in regard to your estimation, if you have neutralizing antibody or binding antibodies that are clearing, that will decrease the physiological circulating level of drugs that will be masked by the presence of the antibodies.
Hmm.
And then the other one that will be really important, right, as we talked about EPO, it's the if those antibodies can be neutralizing and then your drug have endogenous counterpart that is non-redundant, those can be life-threatening situation where the anti-drug antibodies can go by to endogenous protein that is non-redundant, therefore will inhibit the physiological uh, um, activity of that protein. Therefore, those are kind of the key element, right? When we have some subpopulation of antibodies that can be sustaining antibodies, it's still a very big debate within the industry, right? It's antibodies that can bind slightly to the drug itself. It will not provoke clearance, but it will sustain the drug that creates some accumulation of the drug in circulation.
Yeah, check this. You've heard me say this before. The immune system's like this dual-edged sword, and so what Cheikh's describing is brilliant, right? So there's the neutralizing concern, there's the complexing concern, but really the body's so powerful that even if someone kind of, you give the drug once, you maybe give it again, they get this kind of traditional antibody response, and then it can just go away after repetitiveness. So it doesn't really become truly memory, right? And that's like breaking top. Now we're getting into a whole immunology discussion there, but it's almost still, I don't know if surprising's the word, but there's the rate of immunogenicity is that's a debatable topic, but it's impressive how many drugs aren't more immunogenic, but that's a whole nother discussion. But it's not a worry if you generate antibodies necessarily. It's just, again, the most of the side effects will be the efficacy of it, but there is that small part that, if you give it to 100 people and 99 of them develop antibodies and they don't have them neutralizing, yet one person does, that's okay. It's when it's neutralizing over half of the people, or even worse, if it's neutralizing, you start getting into check those IgE responses, right? Other responses that become dangerous. But we'll bring you back to talk about characterization of antibodies. We want to get to the meat of lean here. So maybe how about we start with, we're going to go into our next topic. Just tell us a little bit about the standard of immunogenicity approach.
Yeah, that makes sense.
Yeah, I think the basic of the unwanted immunogenicity testing that we call anti-drug antibody testing, right, is a qualitative assays, right? The reason for that is you don't have your analyte here going to be your anti-drug antibody, but you cannot mimic anti-drug antibody in animal settings. Every single patient can be different, and every single animal reacts differently. Therefore, in that situation, is qualitative assays. They are not quantitative. In that situation, definitely the approach that we're giving to the qualitative ADAs is we have three tiers. Tier one that called screening, tier two is the confirmatory, and tier three is the titration, is kind of summary quantification, and then you can have a final characterization that's going to be the assessment of the neutralizing antibodies.
So to me, as a neophyte, your first step is a yes/no answer. Is there a reaction or not? When you say confirmation, that's like we've done a check and we're saying yes, it definitely is there. And then, like you said, the third piece, the titration, gives you a semi-quantitative take on the degree of it.
Yes, you nailed it. You nailed it, John. Right.
Yeah. Okay.
You are an expert.
Oh, wow. Hey, Cheikh, you broke through John. You turned him into an LBA expert. Finally.
No, I hope in hell of that.
Yeah, absolutely, John. I think
I'm gonna run out and play with a mass spectrometer for half an hour to wash off this stuff.
yeah, absolutely, John. I think the first step is going to be screening and saying yes, no, right?
Yep.
But the process of saying yes or no, due to the qualitative nature of it, if you have a negative, right, as you will be using naive samples to assess that yes, no during your development and validation. If you don't have a signal, it might mean that you don't have an assay, right? In order for us to assure ourselves we have an assay, we are allowing the yes, no to be with 5% false positive, right?
Okay.
We are really at the edge taking the noise of the assay to get a signal, but we know that signal is not specific. At least give us confidence that we have a baseline we can work with, right? If it's truly negative at the first step, then it might be you don't have an assay, you don't know about it.
Sure.
Therefore, why we call in screening, we talk about false positive rate. It's just you detecting enough of noise that give you a signal, but that signal is not true signal against your therapeutic protein, right?
Yeah.
And the confirmatory just is a specificity assessment. I'm saying, okay, I have seen a signal here. I would like really to see if I'm spiking my drug, that is a biologic, within that sample. Am I going to deplete that inhibition, that signal, to tell me it's really toward my drug or something else?
Mm-hmm.
Right? And then the titration, as you said nicely, it's kind of semi-quantification, right? It's just it's looking at the overall dilution, say, okay, most likely, based on the signal I see, relatively, I'm being around this type of titer, right? And the neutralization antibodies, right, that can be either through ligand binding assays or cell-based assays that will, at the end, just to characterize the type of reactivity that you see toward your therapeutic protein.
So this has been successfully implemented for a good 15 to 20 years, Cheikh,
Yes,
right?
yes, yes.
But now that because this segues really nicely into the lean ADA, which you and I are we're kind of aligned on.
Mhm.
But there's also those out there that want to quickly just get rid of 20 years of history, and we're going to talk about that a little bit more. But Cheikh, it's a great segue if you could just it leans right into tell us about what this lean ADA approach is.
Yes, I think lean ADA is kind of very sophisticated term to simply say context of use, right? As I said, right, we is qualitative assays. Therefore, when you do those three steps, they have implications both on timeline, cost, and so forth, right?
Mm-hmm.
Therefore, the industry came to the point, right? Some of us using this in the last, I would say, 15, 17 years, but it came now to the agreement within the industry where people questioning, do I need all these steps, right? And those kind of things have to be on case by cases, right? Lean ADA mean just instead of doing three standard steps, right? The first one that's going to be screening, the second one confirmatory, that will be your specificity assessment, and the last one going to be your titration. If you're not counting the neutralization, do I need all of them? For different reasons, right? That the lean ADA most likely make more sense, right? And really during the preclinical stage. As we know, definitely due to the sequence homologies, right, of different protein, and as the drug now are more humanized, right, we trying to really reduce the potential of having that drug given to human body and that human body reacting with uh, unwanted human response. In the animal, when you give humanize, you're expecting reactivity, right? When you expect reactivity, then you say, okay, do I care about knowing if that reactivity is specific? If, let's say, 90% or 99% of my animals have a reactivity toward my protein that I gave it to them, right? At the time, I said, okay, doesn't matter. It's risk mitigation, right? I can skip the confirmatory to save costs and to save timeline, then go toward screening and I do a titration. I would like to understand just overall what I'm having. And then linking that to the efficacy issues or safety, if I'm seeing some immunocomplex deposition or so forth, at that time I can decide on case by case, right, go toward characterization. Or if I see efficacy issues, and then I can investigate if I need, but that give me yes no question. If I'm seeing efficacy with one animal, right, where you have really a drop in your PK profile, or I'm seeing some immunocomplex deposition or immunopathological issues, right, and then I look back, I see that animal have anti-drug antibodies, I can link together, right? Therefore, I think that lean ADA really approach came in fruition in that manner. Now, in regard to the clinical settings, right, where you have to be most likely, at least where my perspective on it, right, for pivotal studies, I think until further notice, I would do the 3 years, especially if you have drugs that are multi-specific drugs,
Mm.
right,
Yeah.
or drugs that are conjugated
Mm-hmm.
to different molecules.
Mm-hmm.
or even a drug where you see some potential endogenous counterpart, right, reactivity. But again, I think, Dominic, we are part of that. We have a big consortium now that start working across the industry, looking to the need or not or of the confirmatory steps for the ADA characterization.
Yeah, quickly, to kind of summarize a lot, Check, you said that so beautifully. And it's like in the preclinical setting, the non-clinical setting, that you need to use good science to justify going to a two-tiered approach. And there is, justification, but you can't just wholesale say, hey, I'm not doing it. You got to we're scientists, right? You got to prove that you want to make sure that, and you still should always confirm. You can't do a good method, in my opinion, without having some sort of confirmatory or second tier there. So, or even all three tiers, but we can, like, I don't know if concede's the right word, but it often will make sense to reduce your preclinical to a lean ADA. But Sheck made some very good points. Hey, if you're seeing things even preclinically, it's safer to do the threes. And then when we go into the clinic, that's where it really gets controversial, in my opinion, Check, because now you've got, and we've shown some case studies, right? And maybe I'm sure you'll talk a little bit more about it, but just to, like, we hear people in this, some of the members of this consortium say blindly, well, we should just eliminate the three-tier approach. And it makes you and I a little bit uncomfortable, because how are you going to know? We spend all this energy to, like, you just talked about it, getting a 95% confidence, a 5% false positive rate, so that, because if it's all negative, that's bad, right? We've just got years and years of experience and we just want to get rid of it. Again, I'm not saying there isn't a time and a place to use it. It's just, maybe you can elaborate, just tell us a little bit more about your thoughts. Yeah.
Can I ask a little bit? So you have the standard approach is the three tiers. When you go into lean, what you generally taking out of the three tiers? What are people tending to do away with for a lean approach?
Yeah, the tendency is to skip the second one.
Okay.
Yeah, right. And say, okay, what I need to understand, I have a signal, what the semi quantification of the signal is,
Sure.
right? But again, I think it's you don't have one size fits all,
Sure.
right?
Yep.
Where in the industry, I can understand if you have a classical antibody drug,
Yep.
makes sense, right? That is kind of a very easy and so. But when you get
I think when you said, sorry, I didn't mean to interrupt, but when you said earlier that with these more complex designs, the likelihood of immunogenicity increases. Now, my immediate thought was, well, we're doing a lot with like bispecifics and trispecifics, so I
exactly.
assume with those, and you actually then reiterated it. Those are the examples where you really would want to go with the full three-tier approach to be sure you're covered on that stuff.
Yeah, exactly. And again, let just take example of ADCs, right?
Yep.
You can take it as a whole, right? Say, okay, definitely, I'm taking my backbone. I have the linker, have the payload, and so on. So, okay, maybe payload you're not worried about it, right?
Mm-hmm.
But if you talk about between your linker and your backbone in the way, right? Are you interested in understanding where what is triggering, right? The reactivity, or if you have bivalent drugs, right, or tetra-specific drugs, right? I think sometimes just the way we're talking about it is almost we're talking in a very in a vacuum manner, right?
Yep.
Sometimes the drug development organizations they evolving, right? You create a generation of an antibody drug base that can be maybe first in class, right? But your hope is to be having a best in class, right? But at the same time, in your research pipeline, you have new generations, right? Also, right? Therefore, in those situations, sometimes it's good to understand which portion of your drug triggering the reactivity.
Yep.
Even though in silico, at the early stage, you're deimmunizing, you're trying to reduce everything, still, right? Those kind of information, when you do the confirmatory domain mapping, it helps you understand really. Now, it's going to be on case by case, right? If you are a small organization, you don't want to be investing too much money on it. But if you think you have a nice pipeline and that information will be helpful to understand which portion of your tetraspecific or bispecific drug, bivalent, reacting or triggering immunogenicity response, then your confirmatory is required. And secondly, when you have a conjugated molecule, I think from my perspective, it's good practice to have that domain mapping, understanding which portion of your conjugated molecule is triggering your immune response.
Yeah, I want to hammer home that last piece because if you skip a step, right, if you use a lean ADA approach, it's going to be you almost have to restart when you go to do your domain specificity. So it's a shortcut that I would avoid, Cheikh. But again, there's others who would disagree with some of what I just said. No, great stuff there. I think we've hammered home the two-tier approach versus the alternative of 3 tiers. But Cheikh, there's more to this lean ADA, isn't there? And by one more small step back, you mentioned pivotal trials. I think that's a good. I really want to drill in there for a second. What you mean by, to me, I interpret that as if I'm doing a phase 1, if it's a first in human, or even up to my phase 2, I'm going to do all 3 tiers just because it's not a huge investment, right? But then you can make it gives you good data to justify moving away from it, right? Cheikh, is that kind of and when I think about drugs, there is we got to say monoclonal antibodies might have one path, but certainly ADCs and fusions and the more complex proteins, man, I think it would be really bold of you to eliminate a tier before your pivotal trials, and then you can comment on that. But we haven't even talked about cell and gene therapies. We're not going to talk about that today because that becomes, like, to me, a whole nother discussion around immunogenicity and a lean approach. It's really, to me, it's more applicable to the biologic space to date. But
Yeah,
that's
I
my
agree. I think when we get to the ATMPs, right, like cell and gene therapies, I think due to just the low number of clinical patient enrolled in clinical trial, right, that what you're gaining from it.
yeah.
But coming back to the pivotal, right, to me, I think from my perspective, when you do a phase 1, you can skip. You can do lean ADA. Reason for that is usually it's a single dose,
Mhm.
right? Therefore, you don't have this repetitive dose that will be triggering the immune response, antibody maturation, and so forth. But when you go to multiple rising doses, right, in your phase 3, right, in general, or phase 2B in oncology indication, at the time when you're giving multiple doses, right, then you are most likely getting a situation you will be antibody maturation, right, as you're triggering, right, as you're moving along with different repeat doses and different time points. And that can be where most likely I would think at least you can do it now until when?
Mhm.
That can be a question, right? You can do it in meta development, at least understand what's going on, right? And so, or maybe early on, right, in one portion. But at least I think on the situation when you have a bispecific to tetra-specific antibody drugs, or you have conjugated molecule, I think until we get this data out from the consortium, for now I would recommend my client doing that characterization and confirmatory, right?
Yeah.
Right. As again, one of the things that also just to to come back to it, when you do a lean ADA, definitely again, when we do that in our steps, right, in the statistical analysis of the cut point, right, that help us determine the baseline of the assay. In 3 tier approach, we have 5% false positive, right, that will are targeted, right, that can be 2 to 11%. For the tier 1 screening, and then your confirmatory going to be at 1% false positive, right? When you combine both, you're skipping the confirmatory. At the time, your screening now have to be at 1% false positive.
Mm.
That 90%, 99% confidence limit.
Mm. Well, that's a great point. No, that's an important step there that changes it. Just and then I want to be cognizant of time. We've got plenty of time, maybe about 15 more, but I want to really drill into, like, you kind of skirted around it a little bit. Well, not skirted, but like the signal to noise versus cut point determination. Is that, you kind of touched on it right there? But that's not a big change, right, Cheikh? But it is something that maybe tell us a little bit about, like, and again, I want to try to keep it. There's like discovery, right? There's non-clinical, and then you got clinical, right? And so maybe if you want to circle the non-clinical space, because to me the signal to noise ratio, I don't know how prime time this is when my opinion comes in, and maybe it's not the right one. I feel like that's a little bit risky to not use a positive control. But tell us about that a little bit and share your thoughts.
Yeah, I think the signal to noise is the intent is not to not use the positive control, right? Is is to say, instead of assessing a cut point, right? As when we do a cut point, we can have a fixed or floating cut point. But now the consensus is have a floating cut point or floating correction factor. What is behind it is you say, okay, I have a qualitative assay. I can have day-to-day changes and operator to operator changes. How I can have something that will be correcting overall that variability over time without forcing me every single time to be recalculating what I think should be the baseline of the assay, right? When you apply the signal to noise instead of cut point determination, it's just saying, okay, the signal to noise will be applied when you look at, say, if I'm having 5 time points on my day 0, day 7, day 14, day 21. What is important for me is to see that day 0 of that same animal or same patient over the negative control of the assay, what the ratio is, and what the ratio change as you're moving along with the time point. Instead of coming back saying, trying to correct whatever established during cut point assessment, right? I think Pfizer, they published a nice retrospective data, right, in regard to signal to noise, where that retrospective data shows them that their cut point calculation versus signal to noise gave them same output, right? But again, that will be a challenge within this industry, right? In a way, it depends on your assay, right? It depends on the type of a scaffold that you're working with and so forth, right? That you cannot just have one size fits all. It may be on case by cases, right? Based on the data that you understand, and you say, okay, signal to noise makes sense for me. As you know, Dominic, internally, we had this case study that we presented in August where we have not seen one path, right?
Mm-hmm.
Some situation where you have a good correlation between definitely what been calculated signal to noise from the screening versus the titration, and some situation where it allow you or it will ask you to be doing 3 tiers, and others where we presented where we can skip one of the tiers, right?
Yeah, yeah, and it's a lot to unpack in such a short amount of time, Sheikh. But there is you can do retrospective studies, right, where you just sit there and redo the data a couple different ways to determine, and that's what we did. And you showed, I think it was like 60% of the time it might have made sense to drop it, but there was still 40% of the time that argued, hey, you needed all 3 tiers, right? So there's that kind of that's why we want to emphasize that it's not really black and white. But we got a couple more things. Critical reagents check, right? You mentioned that's near and dear to your heart. I love that you mentioned that. I oversee that critical reagent group. No bioanalytical lab. We're only as good as the reagents we're given, right? Check.
Mhm.
We say that all the time. But maybe it's not clear in my head. So I was under the belief that, and maybe it's for clinical trials, you always need a positive control. But in the maybe for if you're doing a lean validation in a non-clinical setting, do you still need the PC? I guess I'm a little. Just share with us a little bit about that.
Yeah, I think the difference is going to be source of PC, right? In clinical, we always recommending to have uh, a generated positive control, right, toward the drug itself, where in the lean preclinical, we can allow ourselves to go source commercial
Ah,
PCs.
I see. I see. I want to say that for the audience there. So
Yeah,
in the clinical setting, you got to make a custom reagent, invest in the reagent, make something specific to your drug, go make that investment. Where in the non-clinical, you can get away with the more commercially available. Got it. Okay. Okay.
exactly. Again, depending what kind of scaffold you're using, right?
Hmm.
If your drug is antibody based, then most likely you can use anything that you can use for your generic PK assays can be used on your ADA for detection, assuming you'll be screening a couple of them, right?
Hmm.
And then some protein also you can have. But when you get to clinical, due to the length of the studies, right? Yeah, and then the need for you to have a reliable source of material every single time, right?
Yeah.
You don't want to be putting your eggs in one basket where you want to rely on commercial outside source.
Yeah.
If that source is kind of stopped, then you have a problem in your assays, or even you don't control the quality of the antibody that been
Yeah.
but that been sold, right?
Especially in phase 3, huge risk if you don't put a campaign together. Major risk there. So actually, I think it's really. Then we can move on from this. What you said was really brilliant. It's like the lean approach does give more like doesn't make it as much of a requirement to invest in that custom reagent. You can do good, reliable preclinical assays, but when we get into the clinic, it doesn't matter lean or otherwise. You got to invest in those the positive control and so forth. So I think that's kind of the take home message I have is like, yeah, sure, the lean approach does allow you to get some, or even I think there's been some advancements in commercially available reagents, even if you do all 3 tiers. But as you get into the clinic, you got to make that investment. And I think the change of if I'm a larger biotech, I don't think it matters. But if I'm a smaller one, it's definitely a huge competitive advantage to be able to get to that IND filing a little bit quicker. Okay,
Sure.
Check, we got a couple more items. John, you want to jump in here? I feel like
No, not at all. I'm fine. I'm listening to it. I'm like I say, it's always good to learn more about other stuff that don't touch at any time. So
yeah,
keep going.
we kind of went over it, but like we're gonna follow our agenda a little bit. When is it suitable to take that lean 80 approach? When should a lean 80 not be used? We kind of went over that, but maybe we jump in there. We got maybe like 10 more minutes here, Shaq. Tell us a little bit about that, then we'll talk about some industry perspectives.
Yeah, I think, as I said earlier, I think definitely in the preclinical stage, it's good to have that question. I think to assess if you can take your ADAs through the lean approach or the full tiers, right? Pending just again, if you have a uh, monovalent type of binding, and then classical modalities where you don't see any high risk for that molecule, right? To give you kind of um, now in the clinical stage, I think where potentially you can use a lean also if that the approach you would like to take is in phase 1, right? Phase 1 is single dose in general, right? You don't have a lot of patients, right? Most likely your ADA incidence should be low, potentially. At the time, you can approach it using the lean just to go through the overall testing quickly. But when you get to phase 2, phase 3, I think now have to be on case by case, right? Based on the mode of action and the scaffold of the drug.
Okay. No, that's well
Yeah.
said. So there is some advantages earlier that you can take, and part of it too, Shek. Let's call it what it is. If you got to rescreen a couple hundred samples, that's not that hard to do, right? It's when you're in a phase 3 trial that if you got to go back, that's going to be really impactful. John, I don't know if I talked over you there or what.
No, not at all. No.
Great. And then Shek, let's just wrap up on like let's what about what's the industry's perspective on it, right? We've heard some, I've kind of alluded it to it. I think periodically we've heard some stuff that's like, wow, that's a little too fast for us. And not that I don't think we're ultra conservative over here either. It's just, it's hard to just ignore 20 years and think, wow, we're just going to randomly do it. We can't emphasize enough that you still should be taking like a, like a real scientific approach. If there's justification for it, then do it, right, Shek? So maybe just share your thoughts there.
Yeah, I think the industry is very interesting, right? Perspective, right? We went through almost a decade where people going really on deep characterization, trying to do the best assays and so forth, and then suddenly we're coming now saying, okay, let's skip this, right? This makes sense, right? It's kind of
like going from like we're gonna do everything 10 we're NAB assays, we're gonna do all of it up front, we're gonna make all now it's like, oh, I'm not doing so that's a good to know.
And what is interesting also, you recall the last meeting we had on this consortium where people are still defending the idea even of having multiple antibodies mixed to have a positive control, right? That doesn't just scientifically doesn't add up, right? Where it's extremely challenging to understand your binding capacity, the displacement of different antibodies against each other, and so forth, right? We one side be defending that, trying to promote that we can have that to have that artificial comparison to the polyclonal antibody, but at the same time also on the lean approach itself, right? Getting maybe really trying to take a kind of a hammer and trying to say, okay, we'll not be doing it instead of getting data from the industry. I would say maybe the more vocal people, some of them will trying to say, okay, let's stop doing lean, let's have it. But I think what you saw on the consortium, I think people trying to say, okay, let the data drive what we trying to
Mm.
do here, right? Let accumulate the data, let
Mhm.
compile data, let test, let look into what the output of that data telling us, and then based on that we can make a recommendation. But from where I sit, I think I'm suspecting it's going to be on scaffold base. Same way what we talk about NAB assays, right? Based on the mode of action of the drug, you can do ligand binding versus cell base. When you need it, you don't need it. I think the lean also going to be on that
Mm.
math, right? I
Wow.
will be having hard time understanding uh, pegylated or
Mm. Wow.
really conjugated peptide. You can approach that without doing domain mapping or domain characterization.
Wow. So we don't even I don't even have enough time to comment on all that juicy stuff you just said around pegylated stuff and cocktails of monoclonal antibodies, which has never been a huge I've never been a huge fan of that because of some of the just what am I looking at in there, right? Kind of mindset. But it's interesting that it's still used. It does. I'm not saying it can't be used. I want to be clear about that. Um, Sheck, any closing thoughts? Because this was thoroughly enjoyable. Any closing thoughts before we're going to put you on the hot seat here? Any last words you
Yeah,
want to give us?
yeah. I think again, I would say just thank you very much for this opportunity and having this kind of exciting, challenging discussion or exciting discussion.
Hmm.
In a way, again, it's just saying at KCAS, right? We
Yeah.
really trying to let science drive the approach, right? We don't have we are agnostic to the approach as long it makes sense when we look at the biology of the and also the scaffold of the drug itself, right?
Yeah.
And the indication.
It's like a math
And
equation sometimes,
yeah,
right? You gotta justify how you got to your answer.
Mhm. Yeah.
So, John, what you want to jump us right into our hot seat questions?
Yeah, I mean, if we're totally wrapped up with this, thank you very much for that. So I will. Should we start with what's your favourite dish to make?
Sheck, you got a favorite meal you like to make or
oh,
something at home? Yeah, no, maybe not you. What's your favorite dish at home?
I know
Whatever.
I am a very barbecue guy,
Okay.
right, and usually when the weather allow, I can do barbecue every Saturday.
Ah. Do you briskets, pulled pork, chicken? Doesn't matter.
Mostly lamb and chicken,
Okay,
right?
lamb and
But
chicken.
yeah, yeah, but
Oh,
I'm very barbecue guys. And
awesome.
then secondly, just a very Asian, South Asian dishes, right? I like them. I don't make them, but
Yeah,
I
you
go
like
out and
yeah, like hot pad thai, or what do you mean by that? Like
yes,
hot Thai
pad thai,
food? Yeah,
everything,
yeah.
yeah, Vietnamese,
Well, yeah, even Indian food. Like the hotter the better, right? Do you like it
yes,
spicy?
yeah,
Yeah.
exactly.
Well, great. I'm hesitant to ask you this one because I don't think you're a big drinker. But do you have a? I feel like I even know the answer to this one. Do you got a favorite beverage that you like to drink, adult or otherwise? I don't know if you got.
No, no.
Not
When I
really.
can, I try to stay on water.
Yeah. No, like in some tea, right? I think you like
Yeah,
the
exactly.
tea. Yeah.
Exactly.
All right. Well, we'll just bomb through that one because John and I like talking about adult beverages a lot.
Oh,
But
sure.
yeah,
Yeah, yeah, yeah.
how about just I don't know if you want to share some weekend
Well,
plans or otherwise.
well, I suppose what we can say here is, yeah, I mean, this is an unusual. Although it's been great to do, this is an unusual time to do a podcast because we're doing it on Monday
Oh
morning,
yeah,
which we
that's right.
never usually do. And
So I guess that yeah,
I'm
John,
not sure.
that question makes no sense.
I'm
What are
not
we?
sure I'm going to be advocating to do it again, to be honest, on a Monday, but never
No.
mind. So, yeah, I mean, it's like, did you do anything much this past weekend, or do you have big plans for coming weekends? Because, yeah, I mean, it's always nice to know. The nice thing about this is that you get an insight to people's lives outside of KCAS, because a lot of the time we don't really get a chance to really dig into what people like to do. So I really like this question in terms of what I mean. Obviously, you're looking forward to the weather getting warmer so you can barbecue. But what do you have anything else that you're having your horizon at the moment?
Is that question for me,
It
John?
Yes,
is.
that's for you.
Yeah.
Yep, yep.
Yeah, yeah. No. Usually, I like really nowadays it's just relaxing, spending some time with the with family, and so forth, right? And rejuvenating for the waiting for Monday, right?
Yeah, no, that's fine, Cheikh. Look, we're right up against time. You've been a wonderful guest. It was just you're just a wealth of information. It was so enjoyable to hear you talking. And I'll end with I'll see you on the funny pages. But John, you can take us out or check us out. But my last statement's always we'll see you in the funny pages.
Yeah, I don't have anything more to add. Thanks very much for joining us, Cheikh. It's always nice to get a little bit of insight into some of the other business directions where I don't really have much to do with them on a day-to-day basis. But yeah, thanks a lot, and we'll be ready for next time.
Yeah, thank you to both of you for having me.
Oh, you're very welcome. It's always a pleasure.